My name is Nadia Jurasevich. I'm a molecular biologist and I worked on small RNA viruses. So I wanted to prepare a brief talk about how to survive current pandemic, and the only way is to learn a bit more about the viruses. So we'll start first with introducing pandemic. This word is created from Greek words pan and demos, which means all nations, and basically it refers to global spread of infection. Pandemic is a global spread of infection. So this was happening throughout all the history of civilization, but I will mention only these two which happened in 20th century. The first one is Spanish flu that happened in 1918, lasted till 1919. It was caused by influenza virus. It killed 50 million people. The reason why so many people in so short time died was that the population came out of the Great War and was really exhausted. The next pandemic in 20th century happened in 1981 and it lasts till this day. This is AIDS caused by HIV virus, 35 million dead. And in 21st century, we had new pandemic with COVID-19 virus, which lasts till this day, and it killed 4.5 million people. This pandemic started in Wuhan in China, and then at the end of 2019, and then spread all around the world. So what is COVID? COVID is an abbreviation for coronavirus disease 19, since it appeared in 2019. It's a viral disease caused by coronavirus, and it causes severe acute respiratory syndrome, which is characterized initially by virus infecting epithelial cells on oral cavity mucosa and nasal cavity mucosa, causing early symptoms of sore throat, cough, and sometimes loss of taste. The virus replicates in epithelial cells, then it spreads further, and it's a respiratory virus, so it can spread to the lungs. Once it infects lungs, we get pneumonia, and the lungs cannot function normally, and there is reduced intake of oxygen and in circulation. So it can be possibly fatal disease with a fatal outcome. So the next question that needs to be answered is, what is a virus? Virus is a small, minuscule particle not visible by naked eye. If it's not visible, doesn't mean it doesn't exist. It does exist on the border between living and non-living matter. It consists of a core where a viral nucleic acid carrying viral genes is packed with nucleocapsid protein. And this viral core is enveloped by lipid layer in which there are few viral proteins, membrane protein, envelope, and the most important protein is, so that was a core, and the most important protein is spike protein. So this protein is very important for understanding how virus affects us and how we can defend ourselves. This spike protein helps virus get attached to the cell receptor, and mediates entry of the virus into the cell. Once the virus enters the cells, it is alive. It can replicate to enormous number of copies, which kill the cells, so the virus can spread further, infect other cells, and then reach internal parts of our organism, other vital organ. Once those organs are infected by the virus, they do not function properly, and this infection can cause severe disease, possibly ending in death. The virus easily spreads between people. So what are coronaviruses? These are small RNA viruses that can infect humans and animals. Better their natural reservoir. Um, the humans get infected via an intermediary that could be either wild animals such as rodents, ferrets, pangolins, or domestic animals such as camels or other cattle. Most coronaviruses in the past caused mild infections, but in 2002, the first dangerous pathogen, which caused severe disease, was, these are intermediary caused, and this is severe pathogens that cause severe conditions and syndromes in people. So these are SARS, MERS, and SARS-2. The origin of SARS is believed to be ferret, of MERS camel, and for SARS-2, it was shown here pangolin, but it's not yet resolved. Once the coronaviruses enter our body, especially SARS-CoV-2, our organism builds defense in order to protect us from infection. The first defense against viruses is called innate natural immunity that we are born with, and it consists of circulating cells such as macrophages and neutrophils, which produce cytokines. These cytokines cause general inflammation in our body, but typically for SARS-CoV-2, this first defense could be harmful to us. It can cause cytokine storm, which means excessive production of these active substances. So this is the reason why infected person has to be treated with early therapy with immunomodulators that inhibit or reduce cytokine storm. These are corticosteroids and non-steroid agents. In addition to innate immunity, we also acquire immunity during our lifespan in contact with infections or with vaccines. So there are two types of acquired immunity. One is passive, which we passively acquire through antibody transfer or as through the transfer to placenta from mother to the fetus. Also, there are active immunity, and this is achieved by infection itself or by vaccination. Infection is considered very risky because immunity is acquired while you're infected and you are not protected during that. Vaccination is a safe way to acquire immunity because you acquire this in advance before infection. So acquired immunity can be divided into active and passive, as I said before. Passive is just a passive transfer of antibodies. Active immunization has two branches. One is humoral and the other is cellular. The humoral is relied on activation of B lymphocytes that are activated after viral proteins from infected cells are presented to B cells. So after they get activated, they start dividing and produce clones. Mature activated B cell is called plasma cell, and it secretes protective antibodies, IgM and IgG. Also, some cells in the clone, some clones remain as memory B cells for later infection, and this is where the vaccines are very important. So we can see at the bottom of this slide coronavirus with all the spike proteins protruding from the envelope, which is surrounded by protective neutralizing antibodies. Another branch of acquired immunity is cellular immunity. This is actively acquired activation of T lymphocytes, which are exposed to infected cells or to vaccine. So these cells, similar to B lymphocytes, multiply, and they create clones. And mature cytotoxic T cell prevents spread of the virus by killing early infected cells. The good thing, so this is cell destruction and memory T cells. So the good thing about memory T cells is that they are long living. They can live for 17 and more years. The other characteristic, they can recognize different epitopes from those that are recognized by antibodies. So they provide additional protection from infection. They also can target conserved epitopes on viral proteins, which are not different among different viral variants. So they also provide protection when the virus mutates. They are cross-reactive with related viruses. So if our defense didn't work and if we are not vaccinated, we end up in hospital. So there are two types of therapies that they use in hospitals. One type of therapy is to alleviate disease symptoms. And as I said before, in this group, there are immunomodulators or such as dexamethasone and other corticosteroids. They alleviate cytokine storm. Then antipyretics are very important to reduce fever. Antibiotics are also used to prevent opportunistic bacterial infection because we are not sterile. Also, other anti-inflammatory substances that inhibit cytokine storm. Also, we have supplements of vitamin C and D and mineral zinc. Other type of therapies act directly on the virus. These are inhibitors of viral polymerase, usually analogs of ribonucleotide units that get incorporated in viral nucleic acid. If you use false ribonucleotide, the viral polymerase is inhibited. Also, there are some inhibitors of viral proteases that prevent maturation of viral particles, so they cannot infect cells. And the third one are synthesized monoclonal antiviral antibodies that can prevent opsonized virus and neutralize. So if all these therapies, so why vaccines? Vaccines are given in advance, as I said before, and also vaccines are a historical milestone in prevention of infectious diseases. Vaccines can prevent and control infectious diseases in the society, and they can also help eradicate dangerous pathogens. So we have two striking examples in the history of civilization and vaccines. The first vaccine was produced in 1796 by Jenner in UK, and this was vaccine against smallpox virus. Jenner used similar virus which infects cattle, it's a vaccinia virus, to immunize people against smallpox. Using this vaccine, smallpox virus was eradicated by 1977. Another example is Salk vaccine against poliovirus, which was developed in 1955 by Salk. This is inactivated virus or attenuated virus. Later, this vaccine was replaced by other vaccine produced by Sabin in 1963. And by 2000, poliovirus was almost eradicated. As this map of the world in 2014 shows, there were 3 zones where the virus was still active. It's in Nigeria, Afghanistan, and Pakistan. So I will talk now about 5 types of COVID vaccines. There are a few other types, but these are the major ones. The first one was developed from inactivated or attenuated virus in China. And there are two vaccines. One is called Sinopharm and the other Sinovac. The next vaccine is produced using adenovirus vector. Adenovirus is the virus which is not harmful, so it's used to carry a spike protein from coronavirus. This approach was used by AstraZeneca, Sputnik, to produce Sputnik V vaccine, and Johnson & Johnson. The next one is protein subunit, in which coronavirus spike protein was used, purified protein, to immunize, used as a vaccine to immunize us against a disease. Then there is synthetic DNA fragment produced by Inovio, which encodes viral protein spike, and also a fragment or segment of viral RNA produced and enveloped by lipid particle produced by Pfizer and Moderna. This is RNA vaccine. So if we compare these 5 types of vaccines to previously produced vaccines, we can say that attenuated virus, as in Sinopharm and Sinovac, was previously used to produce vaccine against cholera, polio, yellow fever, and tuberculosis. Adenovirus vector was used previously to create vaccine against Ebola virus, and protein subunit is used for creating vaccine against seasonal flu, tetanus, and Hep B virus. 2 other vaccines, DNA and RNA vaccines, they don't have any equivalent in all. This is completely new technology. So those vaccines, each of them has immune target, which is recognized by our immune cells once we get the vaccine. So in Sinopharm and Sinovac, when we have the whole inactivated virus, in addition to spike protein that is immunodominant, we have other coronavirus proteins which are exposed and presented to our immune system. For AstraZeneca and Sputnik V Johnson & Johnson vaccines, which are adenovirus vectored vaccines, in addition to coronavirus spike protein, we also have other adenovirus proteins that from the vector that are exposed or presented to our immune system. The problem with this is that, as it said in small letters at the bottom, that presence of some adenovirus proteins may elicit cross-reactive antibodies that may react with our thrombocytes, and this can result in blood clotting in a very small proportion of vaccinees. But similar problem may appear after some hormone replacement therapy and after low heparin dose. Novavax, and it's a protein subunit vaccine, only has one target, immune target, and that's spike protein. The same is with DNA and RNA vaccines. They only present a single spike protein of coronavirus to our immune system. In addition to immune components, these 5 vaccines contain some non-immune components. These are called adjuvants, and they're added to vaccine to enhance immune response to immunodominant protein, which is expressed by the vaccine. So adjuvants are a very important part of any vaccine. I think that there is one vaccine, DNA vaccine, modern vaccine, based on DNA stable molecule that is directly delivered to our cells using electroporation. It's a new technology, and as a naked DNA. Pfizer and Moderna RNA vaccines deal with a very sensitive molecule, so that these vaccines need to be enveloped into lipid nanoparticle. And to stabilize lipid nanoparticles, polyethylene glycol is used, PEG. Polyethylene glycol may cause allergic reaction in some people, but very mild allergic reaction. Just to mention that polyethylene glycol is used in cosmetics as well. So we are now talking about vaccine efficacy. Everybody is just talking about this. I have to mention that all the vaccines that are produced and used these days are designed using a wild-type virus. So meanwhile, there was a lot of mutations occurring in the region of viral RNA that encodes spike proteins. So spike proteins is changed, and those vaccines do not fit absolutely to a new epitopes. So what are the factors that influence efficiency of the vaccine? This is a vaccine type and the dose. Also, its age of vaccinee and his or her physical fitness. So we have 5 different types of vaccines. We didn't have a chance to compare which one is more efficient in a parallel clinical trials. Each of these vaccines was tested independently, and they've been mostly tested on wild-type virus. There are recent clinical trials and testings on a new Delta variant, which is spread all around the world. But I will talk about this in my next talk. In this talk, I will limit my explanation only to some basics of vaccination. So all factors that influence any vaccine is vaccine type and a dose, age and fitness of vaccinee, lifestyle of people, their physical fitness, diet, also virus variant, as I said before. In addition, every vaccine has to be tested in different populations to identify involvement of genetic background. And we know for 2 RNA vaccines, which are based on very sensitive molecule, environmental factors are also very important. I also have to mention that efficacy between different vaccines varies, and we cannot determine efficacy, comparative efficacy, because no one has done parallel experimental trials in which different vaccines are used in parallel. So I may say that scientists and researchers around the world found that RNA vaccines elicit very strong antibody response that protects at the beginning, but the vector vaccines as AstraZeneca, Sputnik, and Johnson & Johnson elicit a strong memory T-cell response, which is very important in repeated exposure to infection. Protective effect of antibodies wanes because they just get reduced. So the revaccination is a necessity. So currently, I'm coming to an end of this talk. Currently, vaccines are the only safe and efficient remedy against COVID virus pandemic. And if we continue to improve existing vaccines so that they can fit better to new viral variants, this will secure lasting protection against the current and similar pandemics in future. And if you want to contact me or ask questions, or suggestions, these are my contact emails. I also want to emphasize that this video was prepared to inform general public about vaccines and their significance in controlling infectious diseases, epidemics, and pandemics. But if you need medical advice, please contact your GP, because the best knowledge of your medical condition is there. And also, when you make your decision, try to be informed, since your decision will have a consequence on your own health as well as on the health of your family, friends, and broader community. Thank you for your attention.